Hello everyone. Today is an important lecture about nervous system pharmacology, an introduction to this important topic. There is no specific reference for the information that will be given in this lecture. However, this is a combination of multiple resources that will introduce the students to basics of central nervous system pharmacology that will apply to all other nervous system pharmacology that we will study later. So this will be some sort of revision of the physiology of the nervous system and transmission of action potential, as well as give examples of some of the pharmacology that used in the central nervous system and how they can modify the nervous system function. To start with, the basic functional unit of the nervous system is the neuron. And the neuron is composed of dendrites, which receives the signals from adjacent neurons, and then we have the cell body or the soma. Inside the soma, of course, we have the nucleus that contains the genetic material, and then we have the axon, which is right here, and we have the axon hillock, which is the first part of the axon, and that is an important structure of the neuron because it sums all the information received through different dendrites, because dendrites are connected to multiple neurons and they receive different inputs from these different neurons. The summation of all of these signals are happened in this important part, which is the axon hillock. Then we have the axon, and then finally we have the terminal buttons. And as you can see, that the signal moves from the dendrites toward the cell body, and then from the cell body to the terminal buttons through the axons. So what is the signal that transmits in this direction? This signal is called the action potential. And to understand the action potential, we have to go back and study the ions and charges that are located inside the neuron and outside the neuron, intracellular or extracellular fluids. So, under normal conditions, if this is a plasma membrane and we throw some ions, and the plasma membrane separates between the intracellular fluid and the extracellular fluid, the ions will transfer equally into the right side and the left side of this graph. Now, but what really happens is that in this plasma membrane, we have some proteins or channels that can control the entrance or the efflux of these ions. Because of these proteins or channels, what happens is that potassium will be at higher concentration in the intracellular fluid compared to the extracellular fluid. Compared to that, to sodium and the chloride which have a higher concentration in the extracellular fluid compared to the intracellular fluid. Now also please notice the charge on each of these ions as you should be expected. Potassium and sodium are positive, chloride is negative. Because of this differential concentration of these ions, what happens is there will be a difference in the membrane potential between the inside and the outside. So the inside will be more negative compared to the outside, which is more positive. And this will lead to a membrane potential of minus 70, and that is called the resting membrane potential. That means minus 70 is that inside is minus 70 compared to the outside potential. Now, in the process of stimulation or action potential, some specific channels will open, and that will lead sodium, as we saw, to go from outside to the inside. and that means positive ions will go inside, and that will lead more positive charge in the intracellular fluid, and that's what we call depolarization or action potential. That will lead to the membrane potential now to plus 40. Of course, as many things in our human body, our system like to go back to the baseline. And to go to the resting membrane potential, now some other proteins that are specific for potassium will open. That will lead to potassium to leave from inside of the cell to the outside of the cell. And because these are positive ions, that will create more negativity in the inside compared to the outside. And that's what we call hyperpolarization, or if it happens after depolarization, we will call it repolarization. And that may lead to minus 70 millivolt membrane potential. To summarize these effects in a graph, let's assume that in this graph, the x-axis represents the time, and the y-axis represents the membrane potential. As we mentioned, the resting membrane potential is minus 70, and that's where the membrane potential happens with no stimulation. If we have a stimulus that is not high enough, did not reach what we call the threshold, represents in the blue line here, if these stimuli did not reach that threshold, they will dissipate and disappear and nothing will happen. They will go back to the resting membrane potential. In case we have a stimulus that reaches the threshold, in that case, even if the stimulus itself disappear, because the neuron reached the threshold, that will lead to the opening of all the channels for sodium, and sodium will influx at higher speed into the neuron. That will lead to the depolarization, or in the graph sometimes they call it the rising phase of the action potential, until it reaches, as we can see here, plus 40 membrane voltage. After that, the cell wants to go back to its baseline, to the resting membrane potential, and that will lead to the opening of potassium channels, and that will lead to the falling phase of the action potential, and sometimes it's even going beyond the resting membrane potential. That's what we call hyperpolarization, because it may reach minus 90, and then before it go back to the resting membrane potential. So the rising phase and the falling phase. So you can guess that what is the ion that is mainly responsible for depolarization is the influx of sodium, and the ions that are responsible for the falling phase of the action potential is the potassium. Let's talk about some notes about action potential. So there is a concept called all or none concept, where, and I already mentioned it, where all the receptors will open and to let sodium to get into the neuron, or none of them will open. As we saw in the previous graph, if the stimulus reached the threshold, it should be all of them should open and let sodium get into the neuron. If the stimulus did not reach the threshold, so none will open for the sodium to get into the neuron. The other thing is that how sodium will get influxed into the neuron. We have two types of channels that sodium can go through. The first one is called ligand-gated ion channels, and from its name, ligand-gated, which means there is a ligand bind to the gate or to the receptor and let the ion open and let sodium go in, or voltage gated ion channels, and that's the voltage will control if sodium will go in or not. So both of them will lead to sodium influx, but the first one, ligand gated ion channels, is through the binding of a ligand into the channel, but the other one is not through binding to ligand, is through actually voltage gated ion channels. And the difference between them also is that it's usually the first step to get sodium into the neuron is through the ligand-gated ion channels, to let some sodium get into the neuron. And that will let the neuron membrane potential to reach the threshold. Once it's reached the threshold, now the all or none concept will apply to the voltage-gated ion channels, and all of them will open and let sodium massively influxed into the neuron. So ligand-gated ion channels, an example of it is the nicotinic receptors that we will bring it multiple times during this lecture. And for voltage-gated ion channels, there are some clinical significance for the presence of these gates, because local anesthetics, for example, and general anesthetics, they use this gate to block, to produce their effect onto the neuron, and thus there is no action potential will fire on that neurons. And it's very helpful when we talk about the decreasing pain during administering local anesthetics, such as during dental practices. Also, there is another clinical significance, which is the presence of tetrodotoxin, which is another example of a drug that block voltage-gated sodium channels specifically, because this toxin, either it's used in research or it's present in some of the animals, especially fish. This is, for example, a buffer fish that is mainly the habitant of the Southeast Asia. And people who eat fish at that area, they are very careful when eating contents of this fish, because they know there is a toxin which is tetrodotoxin that is located inside their body, and this is may lead, it's very toxic toxin, and the eating a lot of this, or sometimes even very little, may lead to a serious or lethal problems to the patient. As I promised, let's talk also about the ligand-gated ion channels, specifically nicotinic receptors, and that's usually the first step in establishing the action potential, where the ligand binds to their receptor, and that's why they are called ligand-gated. And once the ligand is bound, it will open the channel, and now there is a channel that's specific for sodium and sometimes calcium. Since they are positive, so they will make the inside of the cell more positive, and that will lead to a change in the voltage a little bit. That will help the voltage-gated ion channels to open and let massive number of sodium to get in. This is another example of voltage-gated ion channels. We can see under resting membrane potential, sodium is at higher concentration outside compared to inside. And you can guess where is inside from outside just from the negativity inside the neuron. And you can see that the channel is closed because this is the resting membrane potential. But if there is some of the sodium channels get in, and the membrane potential start to increase from -70 to -55, for example, reaching the threshold, that will open the channel, as we can see here, and let sodium goes into the neuron, producing even an increase further in the membrane potential. After the firing of action potential, it will propagate from, as we mentioned earlier, from the dendrites toward the soma or the cell body, and then toward the axon, and then to the terminal button. In the terminal button, we have these vesicles that contain the neurotransmitters that are responsible for transmitting the signal from the previous neuron to the next neuron. So if the action potential, jump toward the terminal button, that will lead eventually to the influx of calcium into the terminal button. Calcium specifically will help these vesicles containing the neurotransmitters to bind to the plasma membrane and release their content of neurotransmitter into the synapse. This is a bigger picture of the synapse. We can see this is the synapse, this is the vesicle, and the neurotransmitter right here. So they go and bind to the post-synaptic receptors, shown right here in green, these structures, and binding to them either will produce excitation or inhibition to the postsynaptic neuron, depending on the type of the neurotransmitter and type of the receptor they are binding to. But all of them, the neurotransmitters, will bind to postsynaptic receptors, producing some effect. What are types of receptor we have? We have two types mainly. We have ionotropic, or what's called ligand-gated ion channels, which is exactly similar to the nicotinic receptors. So these are ionotropic receptors because the neurotransmitter in this case, which is acetylcholine, is released from the presynaptic neuron and travel through the synapse and bind to the postsynaptic neuron, which is in this case nicotinic receptors. This is just an example. We have a lot of examples of ligand-gated ion channels. We have the other type is metabotropic receptors, and in this case there is no channel that is open even after the binding of the ligand to the receptor. And a big example of it, and again we have multiple examples, are called G-protein coupled receptors or GPCRs. The binding of GPCR will not open ion itself. However, it may lead to opening another channels through a second or third or fourth messengers. This is a picture of metabotropic receptors. This is the plasma membrane, and this is the metabotropic receptor. And we can see this is the inside of the neuron, and up here the outside of the neuron. Metabotropic receptors, they are, or GPCR as an example, they are respond to neurotransmitters by directly action of G protein on ion channels, or by formation of a second or third or fourth or sometimes fifth messengers who will target another receptors or channels to open and let change in ion permeability. We don't need at this point to remember these examples. This you can take this in more details in biochemistry or genetics. Probably you know this before. But again, G protein themselves, they will not produce ion influx or efflux from the neuron. And this is the same picture in the previous slide. As we can see here, this is the plasma membrane, and this is the G protein coupled receptors. It's composed of 7 transmembrane domains. It's connected from the inside with 3 subunit alpha, beta, and gamma G protein. It's called G protein because it's connected to GDP. And once the receptor is bound from the outside, it will make a conformational changes in these 7 domains, and that will activate these subunits by exchanging GDP with GTP, and that will let the alpha subunit to separate, and beta and gamma subunit together will separate from alpha subunit. These will activate or inhibit other components in the cytoplasm of the cell, and the alpha subunit, depending on its subtype, it may go activate or inhibit another second or third messengers that may eventually lead, for example, to calcium influx or sometimes sodium influx, or activation of some proteins or inhibition of some other proteins, depending on the subtype, the neuron, and the neurotransmitter. This is also a revision of the receptors we talked about. We can see here that in this receptor or channel, there is no binding site for any ligand. So this is not a ligand-gated receptor. It's actually a channel. And we can see here that it's affected, the opening and the closure of this receptor is by positivity and negativity, so it's voltage-gated ion channels. Compare this to this receptor, we can see there is a binding site, this is the ligand, and once the ligand is bound, it will affect the opening of the channel, so this is ligand-gated channel. This also has a binding site from the outside, and there is a ligand, but on the contrary, it does not have any channel inside it. So this is a metabotropic receptor. We can see that it's connected to G protein, and G protein may affect other ion channels that are not directly connected with the G protein receptor itself. And the more complicated, G protein could activate a G protein, and that will activate an enzyme to build a protein, and then that will produce an activation for another receptor. So sometimes there would be 3 or 4 or sometimes 5 and 7 messengers after binding the ligand into the G protein coupled receptors. Now, we talked about types of receptors. Let's talk about types of neurotransmitters. So the types of the molecules that are released from the presynaptic neuron to the postsynaptic neuron. These neurotransmitters can be classified into excitatory neurotransmitter or inhibitory neurotransmitter. Excitatory means they will bind to the postsynaptic receptor, and they will produce excitation of these postsynaptic receptors by producing depolarization. And inhibitory neurotransmitter means they will bind to the postsynaptic receptors, and they will produce inhibition of the postsynaptic receptors by producing hyperpolarization. So for example, in this graph, we can see here, when we give a neurotransmitter right here, it produces a little bit of excitation. So that's an excitatory neurotransmitter. This is also an excitatory neurotransmitters. But however, you can see that none of them reach the threshold. But if 3 neurotransmitters bound at the same time simultaneously, so that will make the neuron to fire and reach the threshold and produce depolarization of the postsynaptic neuron. So this is an excitatory neurotransmitter because it produced depolarization. Here's again, this is the excitatory neurotransmitter because it produced depolarization. Okay, here for example, this downhill curve, this is due to administration of inhibitory neurotransmitter. So a molecule bound to the receptor, but it produces inhibitory neurotransmitter, inhibitory or hyperpolarization. So this is an example of inhibitory neurotransmitter. Please remember that excitatory neurotransmitter could be ionotropic or could be metabotropic, and inhibitory neurotransmitter could be ionotropic as well or metabotropic receptors. Examples of ionotropic receptors are nicotinic acetylcholine receptors. Acetylcholine will bind to these receptors, nicotinic receptors, that we will talk about next time, and they will produce excitation or influx of sodium and calcium, and that will produce depolarization. So that's why they're excitatory. And because they are ions, they're ion-gated, so there is a channel, and so thus they are ionotropic. Examples of ionotropic inhibitory receptors are GABA receptors. So GABA is the major inhibitory neurotransmitter in the central nervous system. It binds to these receptors, inhibitory ionotropic, because they will let chloride, which is negative, to go into the neuron, and that will produce hyperpolarization. So they are inhibitory and they are ionotropic. But also we have metabotropic receptors that are excitatory, such as dopamine 1 receptors, and we have metabotropic receptors that are inhibitory, which are opioid receptors. So the opioid will bind to the receptor, it will not produce directly ion to go through that receptor, so that's why it's metabotropic, but it will lead to inhibition of the postsynaptic neuron, and that's why they are inhibitory. This figure from Lippincott, we can see that binding to the acetylcholine to the postsynaptic receptors will lead to opening of the channel. And this graph shows the binding of GABA to the postsynaptic receptors will lead to the opening of chloride. So both of them are ionotropic. But one of them is excitatory, which is the acetylcholine, because sodium gets into the cell producing depolarization. And here we have chloride went into the cell, and that will produce hyperpolarization. So how can we differentiate if this molecule is a neurotransmitter or not? So there are some criteria that should be fulfilled by any molecule to be classified as a neurotransmitter. First of all, the localization. The neurotransmitter has to be localized at the terminal button, at the site where it will be released from the vesicles. It should not be in the dendrites, or should not be into the axons, okay, only. They have to be present into the terminal buttons, because that where it will communicate with the postsynaptic receptors. There should be also a mean of synthesis. For example, we have an enzyme called tyrosine hydroxylase that is responsible to synthesize catecholamines such as dopamine. So if we find the localization at the terminal button, we should also look for an enzyme either in the cell or in the cell body or in the axon. There should be an enzyme that is responsible for synthesizing that molecule. There should be also a means of inactivation, because neurotransmitters will be released upon request and should be also stopped at some point. And an example is acetylcholine esterase enzyme, which metabolize or degrade acetylcholine to its metabolites, so the function of acetylcholine will be ended. And it must be released. That neurotransmitter should travel from the presynaptic neuron to the postsynaptic neuron through the synapse. And that's happened, and that was helpful in discovering acetylcholine. When they discovered acetylcholine, they used this simple experiment. They brought a frog heart that is connected to the vagus nerve, which is cranial nerve number 10. And they found once they stimulate the nerve, it will produce bradycardia, or a decreased heart rate. And what they did is that they took the fluid that is supposed to be released by the vagus nerve to communicate with the heart, and they put that fluid into a different heart that is not connected to vagus nerve. So there is no vagus. And they found that when this fluid was applied to this heart, it will produce exactly the same thing as the original heart, where it produced heart rate slowing or bradycardia. So they figured that should be vagus is releasing a chemical that is taken from the area to the other heart to produce the same effect. Also, we should identify the action or the action of that neurotransmitter. In the previous graph, we mentioned that acetylcholine, when applied to the heart, it will produce bradycardia. So there is an identity or identification of its action on different tissues. And finally, we should find the receptor. So the receptor will bind to the neurotransmitter will bind to this receptor in the postsynaptic neuron. We should look for the receptors in the postsynaptic neuron. According to the previous criteria of defining the neurotransmitters, in general, neurotransmitters are classified into acetylcholine, amino acids, biogenic amines, acetylcholine, and we'll talk later and future lectures about this important neurotransmitter in pharmacology. Has two targets or two kinds of receptors to bind to: nicotinic receptors and muscarinic receptors. There are differences between them, and we will study, and we have subtypes of them, and they are differentially localized in different tissues. And all of this is important in pharmacology when selecting drugs to treat some conditions. It is the main neurotransmitter, or it is the neurotransmitter of the parasympathetic nervous system. Amino acids, which are neurotransmitters that are derived or themselves are amino acids. Glutamate is an amino acid, but also is the most excitatory neurotransmitter in the central nervous system. And from glutamate, GABA, which is a metabolite of glutamate, is also a neurotransmitter, and GABA is the most abundant inhibitory neurotransmitter in the central nervous system. So glutamate is excitatory, the most one in the central nervous system, and the opposite is GABA is inhibitory. We have another example of a neurotransmitter that is from amino acid, which is glycine. It is the most abundant inhibitory neurotransmitter in the spinal cord. Biogenic amines can be classified into catecholamines and serotonin. Catecholamines are neurotransmitters that share a common chemical structure, which is called the catechol. Epinephrine and norepinephrine which are metabolites of dopamine, are examples of catecholamines that share this common structure, which is the catechol. You can see it that I circled in the slide right here, and this one right here. So this is the catechol, epinephrine and norepinephrine. We have also dopamine, which is very important in Parkinson and schizophrenia. And this is the chemical structure of dopamine. You can see it also contains the catechol here. Epinephrine and norepinephrine are the major neurotransmitter in the sympathetic nervous system, as we will study later. Serotonin, on the other hand, does not contain catechol, so it's a separate entity by itself. It's an important neurotransmitter in some of the conditions such as depression and eating disorders. Finally, peptides, which are neurotransmitters that consist of multiple amino acids connected to each other. We have multiple examples. Endorphins, which are endogenous opioids, are a common example of peptides neurotransmitter. So now let's talk a little bit about the life cycle of acetylcholine. So acetylcholine is synthesized from choline and acetyl-CoA through an enzyme called acetyl-CoA transferase to build the acetylcholine. From there, it will enter the vesicles through specific vesicular channels and will be stored in these vesicles until the neuron is depolarized, where it then can be released by binding to the plasma membrane into the synapse. After it's released into the synapse, it will bind to the post-synaptic receptors, nicotinic or muscarinic receptors. There should be a mean of inactivation of acetylcholine, and that's the function of acetylcholine esterase, where it degrades acetylcholine into choline and acetate. Acetate will be absorbed somewhere, and choline will be reabsorbed from the presynaptic neuron to be used again for future synthesis of future acetylcholine. This is the life cycle of acetylcholine. I'm talking about life cycle because a lot of pharmacological treatments are targeted toward any of these steps to activate or inhibit this, as we will see later. On the other hand, the life cycle of catecholamine is, most of it is similar, but with different enzymes. So catecholamines start with the amino acid tyrosine, where it is metabolized or transformed to dopamine through two steps. And tyrosine hydroxylase is one of these enzymes to transfer tyrosine into dopamine, which is the rate limiting step of synthesis of catecholamines. Dopamine then transferred into a transporter, vesicular transporter, inside the vesicles through a vesicular transporter, specifically something called VMAT, vesicular mat. Inside the vesicles, if the neuron is dopaminergic, which means releasing dopamine, it will stay as dopamine and it will release as dopamine. However, if the neuron is noradrenergic, which means it should release noradrenaline, inside the vesicles there are some important enzymes that are responsible to metabolize dopamine into norepinephrine. Upon activation of the presynaptic neuron, the contents of the vesicles, either dopamine or norepinephrine, will be released, and then in the synapse it will bind to the postsynaptic receptors, which we have multiple types: alpha, beta, or D receptors, D for dopamine. And the means of activation is either by going into the enzymes into other tissues, or the postsynaptic neuron by two enzymes called COMT, catechol-O methyltransferase, or MAO, which is the monoamine oxidase enzyme, that both of them can degrade catecholamines. Or, which is most commonly, is that these neurotransmitters, before they are metabolized, they can be reuptaken by a specific transporter located in the presynaptic neuron to put them back into the cytoplasm of the presynaptic neuron and then to be stored into the vesicles again for future use, as we can see here. So this is the life cycle of catecholamines in general. So again, mentioning these steps of the life cycle is important in pharmacology because targeting any of these steps will definitely affect the neurotransmitter action onto the postsynaptic neuron by activation or inhibiting depending on the intervention done by the pharmacologist. So let's take advantage from our understanding of the life cycle of catecholamines and acetylcholine to find examples of medications that can target the any step during the action potential propagation. So the first example of pharmacological treatment are drugs that go directly and inhibit the propagation of action potential before it reaches the terminal button and reaches the vesicles in the terminal button. We have, as we mentioned earlier, two examples which are local anesthetics and general anesthetics, which target the voltage-gated sodium channels and they block them, and thus blocking the transmission of the signal toward the terminal button, and it will inhibit the transmission of the signal from the presynaptic neuron to the postsynaptic neuron. So thus you can guess giving local anesthetic and general anesthetic will make the patient lose consciousness or lose sensation of different stimuli such as pain or temperature or touch because they block all the action potentials through the nerves. Some other pharmacological treatment, they target by inhibiting the synthesis of a neurotransmitter, and examples tyrosine hydroxylase, which is the enzyme responsible for catecholamines synthesis. And if we block that enzyme, so then we block the synthesis of neurotransmitters, and that will also affect the function of these neurotransmitters. This is not favorable way for administering pharmacology or targets because this is a generalized enzyme located in many neurons in the body. So if we target this with pharmacology, with drugs, it will affect a lot of tissues and that will produce a lot of side effects. So this is not favorable for researchers to look into this mechanism to find drugs working through inhibiting neurotransmitter synthesis. In the case of acetylcholine, we have the enzyme choline acetyltransferase. But again, this is not favorable because choline acetyltransferase is located in a lot of tissues and a lot of side effects will be combined with this drug if it was discovered by research. Sometimes we can target actually the process of transferring the neurotransmitter from the cytoplasm into the vesicles. And we have one example of such a drug, which is reserpine, which is a blocker for the VMAT that is responsible for the transferring the dopamine from the cytoplasm into the vesicles. So reserpine will block dopamine to be stored into the vesicles, and thus it will not be released. And thus dopamine is used to reserpine is used to treat hypertension in the past, and until now it's used clinically, however at lower rate because many other alternatives have less side effects better than reserpine, and thus it's used only in cases that are unresponsive for other medications. Now we have also some medications that may deal or interfere with the metabolism of the drug. In the case of catecholamines, we mentioned COMT and MAO enzymes. We have a lot of examples that you will study in the future about the importance of these enzymes to be targeted by the pharmacology to treat some central nervous system conditions such as Parkinson's disease, or such as depression and any other conditions. We have some medications that can be interfering with the release or binding of vesicles into the membrane and to release the neurotransmitter into the synapse. Examples, we have amphetamine and catecholamines. Amphetamine can go inside the presynaptic neuron, which is a drug that's commonly used by adolescents, and it can encourage or increase the binding of the vesicles into the presynaptic membrane and increase the release of neurotransmitter, which are the catecholamines, into the synapse. So amphetamine will increase the release of catecholamines into the synapse, and that will produce, you would expect, a lot of side effects. Also, we have another example, which is the botulinum toxin, which is specific for the cholinergic transmitter because it targets the vesicles in the cholinergic neurons and blocks the fusion of the vesicles that contain acetylcholine into the plasma membrane, and thus there is no acetylcholine will be released into the synapse, and that will decrease its function in these synapses. Thus, botulinum toxin is commonly used to treat wrinkles, which are the folds that comes on our skin with age, as well as sometimes it's used for the aesthetic purposes to decrease the muscle tone, because it will decrease the function of acetylcholine. The most common types of a drug that we can use that usually targets the postsynaptic receptors. So neurotransmitters, depending on the neurotransmitter, has receptors in the postsynaptic neuron depending on the neurotransmitter. It was classified to agonists, antagonists, inverse agonists, neutral agonists, biased agonists, and allosteric modulators, and there are many differences between them. This figure shows some of the differences between these ligands. So during resting membrane potential, where there is no ligand bound to the receptor, there is some sort of what's called constitutive activity, that there is this basic activity of the receptor. It's not completely zero. It's actually, there is from time to time, it just fires and produces some activity. So there is a basal activity of the receptor. Now, if we give a ligand that increases its action very effectively, so that's what we call an agonist. If we give a drug that does not do anything to the receptor and keep its constitutive activity, that's called antagonist. And if we give a drug that produces effect in between, so that's called partial agonist. Now, the antagonist itself can be categorized also to neutral agonist, neutral antagonist, and inverse agonist or inverse antagonist. It's the same. If the antagonist did not interfere with the constitutive activity of the receptor, as we can see with this line, so that's called a neutral antagonist because it kept the original or basic function of the receptor. If the ligand binds to the receptor and actually decreases its constitutive activity, so that's called inverse agonist. So we have some drugs that are inverse agonists, some of them are antagonists. In general, they produce the same effect where they do not activate the receptor. And the differences between them, okay, it's more interested in research to find some medications to bind to the receptor as antagonist, but to make the receptor keep its physiological function and its baseline constitutive activity. Moving to the allosteric modulator, if this represent the plasma membrane and this is the receptor where this is the binding site to the ligand shown here, so usually the ligand, either agonist or antagonist, it goes here and bind and produce some effect. In this case, it is an agonist because it produces excitation. In the allosteric modulator, there is another binding site into the receptor, and that's where the allosteric modulator can bind. So it does not bind to the same binding site of the ligand. And we have two scenarios. The first scenario, where allosteric modulator goes and binds and increase the action of the ligand, and that's called positive allosteric modulator. Because positive, because it increase the activity of the binding of the ligand. So the ligand has to be bound to the receptor for the allosteric modulator to work. The other scenario is that the allosteric modulator will decrease the activity of the ligand, and that's called negative allosteric modulator. And again, the ligand has to be bound to the receptor to produce this effect. The biased agonist, on the other hand, is a drug that binds to the outside of the receptor, and it can differentially activate some of the second or third messengers intracellularly and inactivate other second or third messengers. So just briefly, any receptor can activate or inactivate a lot of second messengers downstream, which is intracellular. Now, some drugs that under synthesis can bind from the outside, and they can differentially activate some pathways and inactivate other pathways. That's called biased agonism. Again, it's very important during research to find some medications that can activate pathways that are helpful to the patient and inactivate other pathways that are toxic or produce side effect to the patient. Let's go back to our graph. Some other medications can affect the intracellular fluid into the postsynaptic neuron. And an example of this is the cyclic AMP degradation inhibitors, such as caffeine, which is present in coffee. It targets the postsynaptic neuron directly intracellularly and produce their effect. We also have some medications that affect or target the neurotransmitter degrading enzymes. For example, we have a lot of examples that we will study in the future where they target acetylcholine esterase. We call them acetylcholine esterase inhibitors. If you inhibit the metabolizing enzyme, thus you increase the neurotransmitter action into the postsynaptic neuron. And finally, we have some medications that can target the reuptake process of the neurotransmitter, and that is very specific for catecholamines. We have a lot of examples that block this action or this process. Cocaine, for example, it's a drug of abuse that can block the reuptake of serotonin or norepinephrine or dopamine into the presynaptic neuron, and thus it can increase the concentration of this neurotransmitter into the synapse and increase their action. We have antidepressants, for example. They are specific for serotonin reuptake, and they inhibit this process, and thus they increase serotonin into the synapse and treat the condition that's called depression. This is all about introduction to CNS pharmacology. Please let me know your questions at any time, and I hope you enjoyed it. And I will remind you that there is no specific reference that you find all this information in one lecture. So please feel free to send me any questions you have. Thank you so much for following me.